NAD, isn't a single, uniform pool sloshing freely inside cells. It's tightly compartmentalized. Mammalian cells maintain distinct NAD reservoirs in the nucleus, cytosol, mitochondria, and even peroxisomes, with separate regulatory mechanisms, transporters, and consumption patterns in each. This compartmentalization is critical because many NAD-dependent enzymes, sirtuins, PARPs, CD38, and others, operate in specific subcellular locations and cannot easily access NAD from other pools. For instance, nuclear NAD is essential for DNA repair, PRP1, and epigenetic regulation, SIRT1, SIRT6. Cytosolic NAD supports glycolysis, redox reactions, and sirtuins like SIRT2.
Mitochondrial NAD
Is crucial for oxidative phosphorylation, TCA cycle dehydrogenases, and SIRT3/4/5. Peroxisomal NAD plays roles in fatty acid oxidation and redox balance. A long-standing question in NAD biology is whether different precursors, niacin, NR, and MN, replenish these pools equally, or if some are inherently better suited for specific compartments. NR/NMN claims of mitochondrial superiority stem from early animal studies suggesting faster or greater increases in mitochondrial NAD. After administration, some hypothesize that NR or NMN could directly enter mitochondria, or at least raise mitochondrial NAD more efficiently via the salvage pathway, since NMNAT3, the mitochondrial isoform of the NAD synthetase, uses NMN inside mitochondria. But recent work casts doubt on the idea that intact NR or NMN can cross the mitochondrial inner membrane. Mitochondria lack known transporters for these large, charged molecules. Instead, NAD pools in mitochondria are maintained primarily by NMNAT3 inside the matrix, fed by NMN produced within mitochondria, or possibly by intermitochondrial synthesis from imported nicotinamide riboside phosphate. Meanwhile, niacin replenishes NAD through the Price-Handler pathway, starting with conversion by NAPRT to NAMIN, then to NAD, and finally to NAD
via NMTS.
This pathway operates mainly in the cytosol and nucleus, but once NAD is synthesized in these compartments, it can influence nuclear and cytosolic pools directly, and mitochondria can build their own NAD by using NEM generated in the matrix or possibly importing nicotinamide or other small intermediates. Importantly, studies comparing niacin with NR/NMN in whole organisms suggest no clear, consistent superiority of NR/NMN in raising mitochondrial NAD once the entire system equilibrates. Over longer durations, days to weeks, all precursors raise total cellular NAD to similar extents, with changes propagating to all major compartments through NAD turnover, shuttles, or indirect synthesis. Moreover, whether transient differences in one pool matter clinically is speculative. There's no direct human evidence that brief, compartment-specific spikes in NAD lead to better health outcomes or disease prevention. For example, no study shows that an acute mitochondrial NAD boost from NR/NMN translates into reduced rates of heart attacks, neurodegeneration, or age-related frailty, the endpoints that actually matter for humans. In contrast, niacin's large-scale human trials demonstrate real-world benefits like reduced cardiovascular events, despite niacin entering NAD synthesis mainly through cytosolic/nuclear pathways. Another important point: claims of NR/NMN's preferential mitochondrial replenishment are often based on animal models under extreme NAD depletion, e.g., high-fat diets, genetic knockouts, which exaggerate differences compared to physiologic or mildly NAD deficient states in most humans. Finally, compartment-specific NAD differences shrink over time, since NAD pools interconnect through redox shuttles and continuous NAD consumption/regeneration. After sustained administration of any precursor, the whole cell NAD landscape re-equilibrates, erasing transient compartment biases seen in short-term rodent experiments.